And it's my pleasure to introduce our first speaker today. Dr. Justin Stilwell he's a board certified anatomic pathologist with specialized training in aquatic pathology. He completed his DVM training and certificate in aquatic animal health at the University of Florida in 2016, followed by a combined residency and PhD program in veterinary pathology at the University of Georgia in 2019 and 2021, respectively. In August, he'll be joining the faculty in the Department of Pathology and Population Medicine at Mississippi State University as an assistant clinical professor of veterinary pathology, focusing on diagnostic pathology research and teaching in aquatic animal medicine and diseases. So Justin, congrats on the new position, and I'll turn the floor over to you.
Thank you, Nick, and thank you for the introduction, and thank you everybody for attending this seminar, where I'll be presenting one of the last chapters of my dissertation research on myxozoan infections in catfish aquaculture. So the U.S. catfish aquaculture industry needs no introduction. It represents the largest sector of food fish aquaculture in the United States, generating over $360 million in annual revenue. With global terrestrial agriculture and wild fisheries operating at maximum levels, U.S. aquaculture, catfish aquaculture represents a source of healthy, sustainable, and affordable seafood in a world with growing human populations. This industry produces channel and hybrid catfish and is limited by infectious disease outbreaks, including myxozoan parasitism. So the myxozoans are a diverse group of metazoan parasites infecting predominantly fish, but also amphibians, reptiles, waterfowl, and small mammals. They demonstrate diverse morphology with a variety of characteristic shapes and structural features, and usually cause little pathology in their hosts, except for in cases of heavy parasitism or specific myxozoan species. Of the few myxozoan life cycles described, most are indirectly transmitted, involving a definitive oligochaete or polychaete host and a vertebrate intermediate host. The main myxozoan disease impacting U.S. catfish aquaculture is proliferative gill disease, which was first recognized in the mid to late 70s. It is among the most common infectious diseases of catfish, resulting in high morbidity and mortality in all age classes, though most severely affecting fingerling catfish recently stocked into ponds. Early work focused on the characterization of pathologic lesions, disease transmission, and determining the underlying etiology, which predated the molecular characterization of the myxozoans and implicated various myxozoan species. The etiologic agent of the disease was identified by Linda Pote and colleagues at Mississippi State as Hennegaia ictaluri, with the life cycle involving the oligochaete, Dirodigitata, and the channel catfish. There have been several major breakthroughs in Pgd research in the past 20 years, including the development of a qPCR assay for identifying the parasite in tissues and environmental pond water samples. Variation in host species susceptibility to Pgd infection and disease between channel catfish, blue catfish, and their hybrid cross. Decreased rates of infection and arrested development of the parasite in hybrid catfish, which was later demonstrated in monoculture ponds with lesser Pgd severity, prevalence, and decreasing Hennegaia ictaluri abundance in hybrid catfish ponds. The other major project of my dissertation, which I presented during last summer's AFS Fish Health Section Summer Seminar Series, involved targeted metagenomic sequencing of Pgd cases, which showed mixed infections, differences in myxozoan community composition between channel and hybrid catfish, and lower relative abundances of Hennegaia ictaluri in hybrid catfish Pgd cases. So before showing you some disease gills, I wanted to show you histology of normal gills to give you a frame of reference moving forward. Each gill arch has 2 rows of filaments indicated by the teal arrows, which contain cartilage, bone, connective tissues, and vasculature that supply the gills. Filaments have numerous lamellae indicated by the black arrows. The lamellae are thin, lined by a single layer of epithelium, and contain capillary sinuses filled with erythrocytes indicated by the white arrows. Water passes between the lamellae and the spaces indicated by asterisks, which allows for gas exchange to occur between the blood and the water. This photomicrograph depicts the typical histologic findings associated with PGD, including cartilage lysis and fracture, expansion of the filament by numerous infiltrating neutrophils and macrophages, small clusters of the pre-sporogonic developing stages of the parasite, which are encircled, and epithelial hyperplasia filling the interlamellar spaces, which are indicated by the white arrows. But in addition to Hennegaia ictaluri, there are several other Hennegaia species that infect various tissues of channel, blue, and hybrid catfish, and about 3 to 4 times as many undescribed myxozoan sequences were detected in the previously mentioned metagenomics project, illustrating considerably more myxozoans are involved in these systems than we previously appreciated. The role of these species in Pgd lesions and other subclinical disease is largely unknown, though. So the typical Pgd lesions are associated with these pre-sporogonic stages of the parasite and cannot be identified by histologic examination alone. They require identification by more traditional means, such as examining mature myxospores for morphologic comparisons and doing molecular sequencing. The pre-sporogonic stages in lesions may be lost to histologic sectioning, so that sort of precludes our ability to use techniques like laser capture microdissection for isolating them and individually sequencing them. So we decided to turn to RNA-scope in situ hybridization to definitively identify these developing stages. So for our aims and hypotheses, we sought to identify the pre-sporogonic stages in association with lesions in both natural cases and experimental infections, and assume that they were Hennegaia ictaluri until proven otherwise. Additionally, we wanted to determine the roles of other Hennegaia species in disease pathology, and assume that only Hennegaia ictaluri caused significant pathology during outbreaks, while our alternative hypothesis assumed that other myxozoans contributed additional secondary pathology. So our sequence data for designing these ISH probe targets was limited to the 18S ribosomal RNA gene, with the diagnostic variable region 3 representing the best portion for differentiating species. I developed 9 species-specific targets targeting 5 different Hennegaia species, an actinospore sequence that groups phylogenetically with other catfish infecting myxozoans, and 3 additional unknown myxozoan sequences that we had identified in our metagenomics data set. We had tissues from 4 known pure infections from various Hennegaia species, which we used for partial validation purposes. For our experimental challenge, we used methods as described by Rosser et al. 2019. Fish were divided evenly into 6 exposure tanks and 3 control tanks per fish species. The fish were exposed to water from a Pgd outbreak containing an average of 106 actinospores per liter as determined by qPCR. And these fish were exposed via 6 hour-long immersions per day over 4 days. The gills were sampled from 2 fish per day at day 10 and biweekly starting at week 7. The gills were processed routinely and examined by light microscopy for myxozoan parasitism. And serial sections were made on a subset containing histologically detectable myxozoan development for in situ hybridization. And to examine the roles of myxozoans in natural clinical cases, we examined gill tissues from 7 diagnostic case submissions with previous metagenomics data. Serial sections of 3 representative slides per case were subjected to 1 of 4 duplex ISH assays. 3 slides from an additional case was selected based on the unexpected detection of Hennegaia adiposa sequence data in the gills during the metagenomic sequencing. Hennegaia adiposa is otherwise only known to develop in the adipose fin, so we wanted to see what the role of it was in the gills from our metagenomics data. So these are our validation slides using the known infections of Hennegai adiposa and Hennegai exilis, which were determined by laser capture microdissection and previously published. Hennegai ictalori, which was obtained using a pure actinospore isolate, which we challenged fish to. So we know it was only, these fish were only exposed to one actinospore type. And then a known pure infection of Hennegai adiposa exilis, which Ethan Woodyard has written up and is currently in review. Getting to our challenge data results, this is a table summary of our challenge data quantifying the presence or absence of particular myxozoans. Starting with the pink boxes at day 10, both channel and hybrid catfish demonstrated characteristic Pgd lesions in pre-sporogonic stages, which were confirmed to be Hennegai ictalori by ISH. In week 7 and subsequent weeks, approximately a third of channel catfish each week contained myxozoan plasmodia in various stages of development, while hybrid catfish only rarely contained single very early developing plasmodia. Hennegai ictalori was detected rarely in channel catfish in weeks 7 and 12, with the first plasmodia detected in week 14. And these were present through the end of the study at week 20. Plasmodia detected in weeks 7, 10, and 12 were identified as Hennegai adiposa exilis, which were seen through week 16 and suggests a mixed inoculum during the Pgd outbreak in the source pond. Hybrid catfish contained no Hennegai ictalori signal after day 10 and never formed mature plasmodia at any time point in the remainder of the study. So these images depict the pre-sporogonic stages associated with characteristic Pgd lesions in day 10, along with plasmodia in various stages of development at weeks 14 and 20, confirmed to be Hennegai ictalori by ISH. These images depict developing and mature plasmodia identified as Hennegai adiposa exilis, with early plasmodia forming on the lamellar tips and eventually filling the interlamellar spaces. So for the naturally infected clinical cases, these are stacked bar charts of our metagenomics data set depicting the myxozoan communities of each case with percent relative abundance on the y-axis and case number on the x-axis. The blue boxes indicate the selected cases from channel and hybrid catfish Pgd cases for ISH. The asterisk indicates the one case that was selected for Hennegai adiposa singleplex ISH. This table summarizes the presence or absence of particular myxozoans in each case, and similar to the experimental challenge, pre-sporogonic stages associated with the characteristic Pgd lesions were consistently present and associated with Hennegai ictalori, which was confirmed by ISH and indicated by the pink box. The presence of other myxozoans in various stages of development was common, though appeared random from case to case. These myxozoans were often associated with secondary pathology such as inflammation, hemorrhage, or epithelial hyperplasia and filling of interlamellar spaces, depending on the stage of development, of myxozoan development. The red box shows at least 5 to 7 different myxozoan species were detected by ISH in channel catfish Pgd cases, while only 3 were seen in the hybrid case, confirming the mixed infections identified by metagenomics. Although not identical to Pgd pathology, the pre-sporogonic stages of other myxozoans rarely produce Pgd-like lesions. So panels A and B in this figure depict multifocal chondrodysplasia throughout multiple filaments. The panel C illustrates predominantly lymphocytic inflammation, epithelial hyperplasia, and lamellar fusion, though the gills are heavily infected with ichthyobotonecator flagellates, complicating the attribution of disease pathology. Multiple multinucleated pre-sporogonic stages were identified as corresponding to the rabia type TGR 2014 actinospore sequence and were widely disseminated in some of the gills from this one particular case. Other gills from the same case, though, contained presporgonic stages of Hennegai ictaluri and more typical Pgd lesions, though. These are photomicrographs from the case containing the Hennegai adiposa sequence data, which show lymphocytic infiltration, cartilage degeneration, and a multinucleated presporgonic stage in panel A, cartilage lysis and presporgonic proliferation in panel B, and cartilage fracture and presporgonic stage release in panel C. Panel D confirms the identification of Hennegai adiposa by ISH and suggests Hennegai adiposa may use the gills as a portal of entry and site of early replication prior to reaching the adipose fin, though this requires experimental confirmation via challenges with a pure actinospore isolate of Hennegai ictaluri. So we can't say for certain just yet that this is Hennegai adiposa. So the major findings from the experimental challenge demonstrated differential susceptibility between channel and hybrid catfish to infection and development of Hennegai ictaluri and other myxozoans, with arrested development and or improved clearance of multiple myxozoans in hybrid catfish, which is consistent with previous challenges by Rosser et al. In 2019. We confirmed the mixed infections previously detected in clinical Pgd cases by metagenomic analysis, as well as mixed infections in the experimental challenge. The identity of presporgonic stages associated with the typical Pgd lesions were confirmed as Hennegai ictaluri in both clinical and experimental Pgd infections, though other myxozoans appear capable of contributing either secondary pathology or rare Pgd-like lesions. Future directions should look to exploit the influence of fish host susceptibility to infection by developing optimal crop rotation strategies, which would involve alternating between channel and hybrid catfish in such a fashion as to prevent any one particular myxozoan from dominating in a pond and thus potentially mitigate disease outbreaks. More work is needed to investigate methods of myxozoan dissemination and persistence on farms, given that hybrid catfish appear to have considerable resistance across multiple myxozoans based on our findings here. Determining the clinical significance of these known and unknown myxozoans will require uncovering definitive hosts, identifying actinospore sequences, and maintaining parasite life cycles in the laboratory. And so using these pure isolates of actinospores, we can examine specific tissue tropisms, as well as the roles of myxozoans in causing unique pathology and disease specifically attributable to them and not to a mix of species acting together. So at this point, I'd like to acknowledge many people who've contributed to not only this project, but to my training over the past, man, it's been a long ride. So I think, wow, 13 years at this point, including undergrad. So I want to thank my committee, Drs. Al Camus, Matt Griffin, James Stanton, and Nicole Nemeth, our lab technician, John Leary, the folks at MSU who have been with me on my dissertation throughout multiple projects, including this one, including Lester and Graham, Ethan, Mackenzie, Adrian, Brad, the UGA Histology Lab for cutting thousands of slides for me over the years, the UGA Department of Pathology for their constant support, and for providing me the opportunity to be here, and my wife, Natalie, and my parents, Cynthia and Winslow. And these are my references, and I'll be happy to take any questions.
Thanks, Justin. Great presentation. Folks, feel free to put some questions in the chat box or unmute yourself and ask them directly. And while they're doing that, Justin, you got some congratulations on the new job in the chat box here and compliments on the images in the presentation.
Thank you.
Go ahead, Ben.
Oh yeah. This is Ben from OSU. I was curious what the gene was for the actinospore sequence you used. What gene was that?
18S ribosomal RNA.
Okay. Have you looked at all into like I guess actinospore specific probes?
So I mean, the probes can be made to actinospore sequences just as easily as myxozoan sequences. It really just comes down to how different the sequence data is, and so we had to pick a region that had enough dissimilarity to allow us to distinguish between different species, and so that diagnostic variable region 3 had just enough dissimilarity to allow us to design these unique probes, and so yeah, I mean, we were able to design a unique probe to that actinospore sequence. And so it's not necessarily detecting actinospores, it's probably detecting the pre-sporogonic stages and myxospore stage of that one, but that the associated myxospore stage for that actinospore has not been described yet. So that's why I'm referring to it as its actinospore sequence and not the myxospore.
I see, right on. Thank you.
Yep.
Hey, Justin, this is Jeff. Quick question. How different are those probes that do you not get any cross reactivity across the different isolates or different species?
So that probe sequences have to be at least 80% lower than 80% similar. So that's why it sort of narrowed our region of what we could use. So basically, these RNA scope probes usually have many different combinations that spread across like a 5 to 1,000 base pair segment of a gene target. But we were able to use basically each of these probes only has one identifying combination, and we were able to do that because the 18S ribosomal RNA gene is so heavily expressed within the organisms as well as in the genome that we didn't need a whole lot of coverage over a particular gene. We just needed one area, and we just had an abundance of the gene target in the samples. So,
all right, nice, thank you.
Yep.
Go ahead, Esteban.
Hey, Justin, great job and congrats on the new position. I was wondering, you mentioned that the hybrids and the blues, have different susceptibilities to some of these parasites. And if I recall, with Pgd, much of the damage is basically blame on the immune response, that the catfish, that the channel mounts against the Pgd. When, I don't know if you have, or if anyone else, have challenge, against these guys, is it really that infection doesn't take place, or is it that basically the immune response act different, between these different species, again, more based on your findings with the in situ and the inflammatory response?
That's a great question, Esteban, and that's still up for debate. As far as whether or not the damage is the immune system versus the parasite, I know people in both camps of that argument. I know there is a paper that Jan put together with folks at PEI that showed early cartilage degeneration in the gills prior to the parasites arriving there and prior to the inflammation arriving there. They basically did an ultrastructural examination of those gills. And then as far as whether or not it's clearance, whether or not the host is able to recognize it, or if it's just the parasite not as well adapted, I think there's still a lot of questions about whether or not it's parasite or sort of host, parasite recognizing host or host recognizing parasite. I think there's still more work to be done with Pgd specifically. I think they're starting to answer more of those questions in the Mixobolus realm and the Ceratogenopa Shasta realm. They're starting to look more in the transcriptomics of the immune responses and looking at some of the genomics of the parasites to see if there is any sort of host parasite interactions. But it's definitely a area that I'm interested in pursuing continuing PGD research at MSU. So that would be something to look into for sure.
And in those, if I can ask you just the last one, I promise, because I know that Al is already yelling at me. But I was wondering, Justin, if when you read all of these histological slides, natural cases, with these henogaias, I'm guessing you're finding also a lot of co-infections with different bacteria. I know that you mentioned that for some PGD, you find co-infections with some of these other henogaias that are like hiding a little bit. So I was just wondering, with this intense inflammatory response that you're having, do you find more or less, co-infections with bacteria? Because a lot of it could be actually non-specific and fighting off, the bacteria. So I'm just curious, how often do you find co-infections in these gills?
So I know they see co-infections with things like columnaris pretty frequently. Histologically, histo is not always the most sensitive means of identifying bacteria. So they usually have to be there in pretty heavy numbers for us to see them in a histologic section. I mean, there are other parasites in these ponds. There's a lot of different pathogens going on in these ponds. So I'm sure there are co-infections, and I know grossly the diagnosticians will see co-infections with bacteria and other parasites. So it's definitely another factor. And I mean, I had the one case in this presentation that had ichthyobodo covering the surface, and then the myxozoans were in the filament itself. So I mean, I think there's a lot of stimulation of the immune system going on there for sure. I think the PGD pathology is pretty specific to that though, or pretty unique to that. I think the parasites and the immune response are pretty characteristic for that, and that I don't think that necessarily relies on a co-infection of other myxos or other pathogens, but you can have co-infections that make things worse. They contribute secondary pathology and make things worse for the animal.
Thank you, Justin.
All right, thanks again, Justin, for the great talk. Virtual round of applause for you.